first of all, go through the implementation synthesis slides, and then split into small groups to flesh out. And we need to identify champions. And these champions will be involved in modeling in the data integration workshop, and the standardization and intercalibration workshop, the informatics and data management workshop, and also in education and capacity building. And I think you already know about the online form that is available to fill out, to volunteer, to do these activities. But to take this forward, the next steps, that's what we need. We need volunteers to do these. We need champions who are going to help do all this. So I think, unless there's anything else, we can move straight on into the implementation synthesis slides. So who's the leader? Who's going to?
So obviously, I'll review the implementation slides that we put together. So as a reminder, just to bring you back, we were talking about time, space, depth, and lab implementation. So for the time, the major takeaways here were obviously that leveraging time series, I think this has come up quite a few times, that we should really try to, BioGeoScopes should try to leverage what's already in place. But then we need to really consider which time series should we target, which regions of ocean have the highest variability and need to be sampled more, right? And so we need to balance sampling with thinking and analysis, that's an answer. Okay, so different regions of the ocean have different time scales that need to be considered, right? So thinking about if currents move rapidly, that we can't separate space and time as much as we'd like. The times of sampling need to be well documented, and the metadata need to be organized. And the international community of BioGeoscapes should have a mobilization network ready to respond to the extreme or unusual events. And that was something that folks really took to, is this idea that we have a rapid response team that could respond to disturbances or whatnot. Okay, so then in this idea of space, where we would like to sample, the areas to target. OMZs have come up quite a few times. Then there's also considering islands, polar regions, eddy systems, river plumes, vents, shelves, upwelling, coastal areas. So a variety of different specific sites that could be considered. Why feasible, and what is the value of these international collaboration efforts such as BioGeoCase? So again here, capitalizing on what's already in place through LCR, the time series. Also using real-time data, remote sensing, and higher resolution to target. So I think maybe a little bit of leveraging things like Argos, satellites, and gliders. And we can also use the BioGeoscapes then to really leverage and grow the shared expertise to build a database across groups and minimize competition and maximize collaboration. Okay, so what do we need to put this in place? So new platforms or sharing, right? So new mechanisms of sharing across things like our time series data, international data management, and networking must include funding agencies from the start. And then this has come up a couple times, of thinking about the ethics, right, of using networks to engage regional sciences and have everybody be a part of the process. Okay, so this is a virtual one then. We didn't get to talk with these folks directly, but just to try to summarize really quick, thinking again about sections, right, surveys versus processes. What are the levels of measurement that we could divide that up through? So level 1 being biomass, chlorophyll, hydrography, whereas level 2 you could be defined specifically for BioGeoscapes. And then they're going through and prioritizing which of these levels we would go through, right? And so you can, I'm not going to go through and read all the measurements, but you can see the level of prioritization for things that are like primary nutrient analysis, primary productivity, respiration, and then moving up to genomics and all the way up to level 3, metabolomics, proteomics. Okay, the transects would really focus on open ocean with gradients in pH and temperature from the poles to the tropics. The process crews would be around islands, upwelling regions, and music scale features. In time series, could we think about, getting at many coastal regions around the globe, again, augmenting the existing time series. So one question is, are there areas where there are no time series that we could leverage as biogeoscapes to really kind of focus on, right, and facilitate that new development in some kind of gap knowledge? And then just as it's come up a couple times, think about carbon sequestration, right, as an important communication to stakeholders, really, again, think about this new interest in CDR. Okay, so then with depth, what are the things that are moving in terms of thinking about vertically with the biogeoscapes goals? Well, the adaptive sampling strategy, right? So that we can go out, we can really survey the water column and then ensure that we're getting the right depths for the biogeochemical transformations that we're interested in. A goal maybe to increase measurements below the euphotic zone will help us with these kind of depth scales and time scales of change in the deep ocean. And then a kind of a desire or want is to actually have in situ or pressurized incubations to get at rates for the whole ocean. Okay, so what can we do again in terms of depth with a program like Biogeoscapes? We need recommendations for how to target depth features and resolution. So I think what that's going is that we could have an adaptive strategy, but what are the things that we're trying to adapt to? Don't waste the water, which was a really interesting one. Is like, work hard to get that deep water sample, and so it's really a shame when it goes overboard. So can we have things in place that are going to leverage that, right? You could use that opportunistic sampling. And then making use of the autonomous and untethered samplers to increase the capacity for deep water sampling. Okay, so then thinking about, okay, in terms of laboratory tools and experiments, really kind of there's this kind of circular idea here that we can use field observations to prioritize lab experiments, which are needed to parameterize models. And so just to sum that up, obviously, we need to communicate among one another. And there's a variety of lab experiments that have been proposed in these groups, actually making the rate measurements in the lab so that they can be calibrated, trying to dissect microbial interactions in the lab, getting at the key elementary stoichiometries. What is the organic matter composition that's coming off of things like phytoplankton or being taken up by bacteria? This really grand challenge of increasing the amount of genes that have a functional annotation, and then defining biomarkers. And then there's this question of, do we need new model systems that are really kind of capturing the important biogeochemical processes in the ocean? There was a lot of talk, this was my group that I was in, so of really having a way to communicate these results. So we all know that we can put our sequences in databases, but it's kind of clear that we don't always know when somebody else does something new, right? There's a new genome available or a new transcriptome for the eukaryote folks. And so having some kind of biogeoscapes communication database where we really know what's going on with each other in terms of these lab experiments. Training opportunities. So one thing to think about is, could we have a summer school to train students where we train them on the protocols that we're going to use? I think this extends beyond the laboratory to thinking about the cookbook, right? And so that we have a group of folks that really know how to do these protocols the same way. And then standardizing our workflows, not just with the rate and stock measurements, but things like genomic analysis. And then facilitating networks to democratize capabilities, right? So I think this has come up with a lot of people that, do we need a place where we can send our samples, things like a central source where we can look at trace metals, DOC, omics, rates? Is there a place where we'd want to do kind of collaborative mesocosm experiments? And then, do we want to have this idea of having a high throughput standardized QC methods across groups? Okay, for models then, for the data, we want to make sure that the data, follows these fair practices with the need of distributed semantic approaches to share and be used by all disciplines, okay? Again, that's just getting on that communication idea. We went through the types of modelings, right? This was actually kind of a really instructive thing for those of us not directly involved in the modeling. Really thinking about, what do these two groups of models tell us? We have the predictive models, right? and then the synthesis models. And how can we best leverage our sampling and our experimentation to really kind of leverage the power of each one of those different types of models. And then again, the usage, thinking about how we're going to use the models. I think this is really tying into like bringing people in from the start and thinking about experimental design, hypothesis testing that's coming out of the models, what kind of scenarios can we kind of simulate, and then extrapolating into the future, or leveraging models to kind of do that. The opportunities then, this was an interesting idea that came up, was that could we design a biogeoscapes modular modeling system that leverages all these things above and moves beyond just validation pools and processes and develop a distributed modeling environment. So I think I don't want to say too much because you know, this was another group's idea, but the idea was maybe that we could develop a modeling environment that's accessible to much more than the specific modelers, right? And you could plug in your data into a model that fits into some global model system. So it was a really interesting idea, and I'm sure the folks that focus on that would be glad to talk more about it. Good. That's the last one.
Thanks. Ben Twining, Bigelow. Going back to the platform that was, I just wanted to ask a bit more. Like, when you say we need a common platform, is that just a sort of a data, you know, not only hosting, but sort of visualization platform? Or what was, it was maybe one slide further back. Anyway, anyone from that expand on that at all? No, maybe one further back. Sorry. Maybe one further back. There, new platforms or sharing. I didn't quite know. Do we mention that as a, like so again, being familiar with GeoTraces, there's eGeoTraces as a way to visualize some of the data before downloading it. Is that the kind of thing that is meant by this? Or just a place to have like all of the sequencing or data together, or that farms it out to other places? Anyway, just curious about that. Okay, this is just like a sharing platform, not a data hosting platform. Great. Just wanted to show that. Thank you.
Great. So I think it's been really interesting to see how all these different synthesis groups have come together, and how these topics have sort of worked out. We're now going to move on to fleshing out the science plan. I'm not so familiar with Google Slides. So our major task this morning is to flesh out this science plan. So how we're going to do this, there is a straw man science plan that you can reach on this tiny URL link at the bottom. BGS Science Plan. And we're going to sit in groups, theme groups, so theme 1, theme 2, and theme 3, and work on these science plans. So you self-allocate to groups, and you don't need to stay in the group. You can move from one group to another. So we want your expertise. We want, we want that information that we all have. We want to harvest that to start fleshing out this science plan. The Strawman Science Plan at the moment is structured in a way somewhat analogous to the GeoTraces Science Plan. So we have these headings, current understanding for each topic. And I think this is what we're going to focus on mainly this morning, these themes and topics. So we want information about the current understanding. Doesn't have to be fleshed out in great detail. What we're looking for here is key papers. So if you think a paper really speaks to this theme, drop the DOI in and write a little sentence summarizing why you think that paper is key to this particular theme or topic. Use the breakout summaries to populate the areas of advance. There's lots of information from the breakout summaries about areas of advance, so use those to populate that heading. We need objectives, and here we're looking for quantitative goals. So sentences start with those nice objective type words like identify, determine, characterize, and avoid sort of non-quantitative terms like generate understanding, things like that. Perhaps that's my personal, but I think it's good. So, but keep them broad scale. We're not looking for a great big list of really tiny objectives. So, broad scale objectives. And again, use the breakout session summaries to help focus on those objectives. So we've just had our implementation summaries. So think about which of these key approaches are the best for addressing the particular topic that we're fleshing out. So is it going to be a combination of field lab or models? Think about how it will work for this topic. And then finally, perhaps simply, what other programs do you know about which are also addressing that topic? And let's, generate a list of other programs to interact with. We're just looking for bullet points, short sentences. So, but also when you're doing this, think about the story and the way that the science plan will flow, so that we've not got random bullet points sort of all over the document. So try to fit your bullet point into a sort of place that makes sense in terms of the other bullet points that are present in the document.
Hi, I was part of one of the synthesis groups for one of the themes. And one of the things we struggled with was where to put the science questions that we've been so focused on within the structure. So maybe some clarity there so we have some consistency of like,
in the areas,
science questions.
Great, that's a good question. So I think I'd put those in the areas of advance. That's where the science questions go. Yeah. Any other comments or questions about this?
Hi, Martha, over here.
Sorry.
Yeah, just to add, as people are logging in and seeing, you'll see that the notes from the slides that we synthesized from are pasted into this outline already, but it's a bit of a mishmash. So that's what we mean when you say draw from the groups. You can actually, it's in the outline already. You can go back to the original documents too, but to make it easier, there's a lot of information there. It just needs to be organized and prioritized and stuff like that.
Synthesized in some cases.
Can I make a comment? In Theme 2, we did, the areas of advance was like very general, and we put the questions in the objectives. So if we want to change, then Theme 2 has to, So we are going to agree areas of advance is where we put the question, and then the objective, we create a new objective.
So the other thing in the straw man is it's about also the overall structure of the science plan. And I think if you have particular ideas about how the structure might work for biogeoscapes, then please comment that in so that we can develop a unique biogeoscapes structure, which also covers all the bases that a science plan should cover. For example, I think one of the things that has been coming out of this meeting for me that I sort of noticed was about the implementation and this franchise model. So I have suggested in the straw biogeoscapes plan that this way that we're going to implement biogeoscapes is kind of up front before the topics. And so that means in the implementation part of the science plan for each topic, we don't have to explain everything every time. It can be just referred back to the different kinds of franchises that might be applicable. in this approach. And please comment on this. You know, these are just ideas. So, so if you see like comments that people have added, review them. You know, feedback on those comments. Anything else? Next slide. So this is a dense slide. So these are the themes. So the priorities are themes 1 and 3. Theme 2 was a little bit more structured, but themes 1 and 3 really need to start populating. Let me just get rid of that box. Populating the headings. So these are the topics and the themes, and there's also at the end an international capacity building and collaboration, intercalibration activities, data management, and informatic pipelines. So again, if you have particular knowledge relevant to those topics, please, you know, populate those topics with that knowledge or those ideas.
Martha, can I add to that? So we're going to have future meetings and probably reports for intercalibration activities and data management and informatic pipelines, but it'd still be good to have brief chapters in the science plan describing their importance and, you know, potential future efforts and things like that. So feel free to go there and populate those as well. But keeping in mind that they don't need to be expansive because there'll be future whole workshops on them.
Okay. So we're going to position people in different rooms. Heather?
If you go back to the first slide, I put room assignments there. So Clark, theme 1 is going to be right in this room. Theme 2 is going to be right outside this room in that little nook behind the coffee, right over there. And theme 3 is going to be in Clark 509. And you're all working in the document.
Yeah.
You have 1.5 hours. You have 1.5 hours to do this.
And you can have coffee.
Martha, hi.
Let Al speak. Going to tell me something.
No, can you go to the next slide with the sort of subtop? No, the next one. You know, to what extent did you consider the fact that some of these subtopics are really kind of general organizing concepts for the whole program as a whole, right? So for instance, microbial interactions in a 4D ocean, yes, it was discussed underneath theme 1, but in many respects, that's a whole organizing concept for how we want to even start to consider to study these questions. The same could be argued for the modeling, you know, the key gaps. Do those gaps really only pertain to the topic under theme 3? I just wonder if that got discussed, even coastal, you know, some of the coastal, mesopelagic. You know, are we really envisaging a textbook type science plan that goes through, you know, upwelling zones, OMZs, etc.? One might imagine those being sort of higher level sort of framing components of the science plan and not necessarily wedged under a particular theme, because it sort of, you know, the coastal ocean, you could argue, cuts across all of these 3 themes. Did that get discussed?
No, it hasn't been discussed, but I think it's an excellent thing to add in when I mentioned about thinking about the structure. So this is exactly the sort of thing that we should be looking at in terms of the structure. So are these themes, we don't need to have 6 themes under each topic.
6 topics under each theme.
That's the one, 6 topics under each theme. You know, we want topics under each theme that make sense, that are cohesive. So yes, please put these thoughts and ideas down at this point in this science plan. So in this straw science plan. That's why it's a straw science plan, because it's there to be broken, right? So I think add these ideas, yeah.
I think it's worthwhile noting that, you know, these were breakout topics. These weren't necessarily meant to be science plan chapters. Yeah. So we might need to do some merging of some of these. We might need to eliminate some of them. Some of them are high-level overarching principles that are really pushing the whole idea of biogeoscapes forward. maybe we missed the boat completely and missed topics, important chapters that should be in here. So that's really the important work of the synthesis, is to take all of these different breakout discussions and turn them into an outline.
Can I make a suggestion? I think this is really important, like how this is really was not meant to become like the science plan. No. They were really to provoke discussion and, you know, to brainstorm. So I feel like, can we make a group right now that is going to work on this? Just brainstorm together what will be the best way to maybe like to follow up with what Al said, but instead of having just Al write a little comment, is it worth it to have some people right now that will sit and think about this together? Is that worth it? Al?
So how many people would join that group if we had a group like that? Okay, that's a relatively small number. So let's have a group like that, and then other people can go off into themes and topics.
The only
thing is that, but it doesn't matter, because all that information is, if it's just a question of moving it around in the document, you still got that information. So we still have a synthesis of what we've discussed this over the last 3 days. We've still got all that information. So we don't have to have, I don't think we have to have the structure finalized before we start fleshing out the information.
So we could call that the structure and what are we missing group or something?
Yeah.
To have another parallel conversation next session.
And maybe we need another online. Where will that group meet? Yeah, where will that group meet?
Sorry.
50.
While you figure out the logistics of where that group might sit, I just wonder what the charge to that group is. Is it to look at these subtopics under each theme and really take a fresh feel of kind of, are these actually cross-cutting general organizing? You know, if I look at theme 1, I feel like actually the last 4 of those could all be generalized organizing principles for the study, right? Do you want us to go through the themes and do that? Kristin, do you have an idea?
Yeah, I don't know if it's a good idea, but I was thinking, I think in parallel, because I was reading through, and what struck me was that there is a lot of overlap in the way that these things were addressed in the subtopics of theme 1 and 3, and theme 2 sort of managed to get above that, and it makes sort of more sense. So that's where I feel like I would want to start, is like just brainstorming. You know, where do we as that group see like things pulling out? Okay. Right behind you.
Thank you. This is Kazi Vogel, University of New Hampshire. Well, I don't know how you write proposals or documents like this, but I feel like, and I think it's a great idea to start on the objectives for people to working in this. And I think if we have objectives for the different themes, then from those objectives, naturally, there will be topics emerging. So we have 90 minutes, probably 80 minutes now, because we've already talked 10 minutes. So if 80% of this group will sit down and write down objectives, and the other 20% try to organize the topics at the same time, I feel like it's a waste of time.
Right. Okay.
I think let's start with the objectives, and then.
So you think 100% of the people should be writing the objectives?
Yes.
Okay.
Okay. Got a live one. So I'm Eric Webb. I'm at the University of Southern California, and I was looking at the big science plan document and actually worried quite a bit about what you were just saying. And Allison at 9:30 or 9:27, Allison Santoro said, I have a lot of trouble distinguishing between theme 1 and theme 2. As written, I would suggest simplifying these themes so they could be easily understood by anyone, a funder, politician, general public. they're very jargony. And when I think back to the first day, I thought there's an awful lot of overlap between 1 and 2. I worry if you have a group like restructuring, that other people are going to be wasting their time. I kind of think, I agree.
So in that sense, you're suggesting that we concentrate on pulling out the individual objectives that we want to address, and then we put them into structure and into the themes.
Just to add, that might make sense. Just given, we only have about an hour or so to do this, it might be hard to, but, maybe a group after the fact could help synthesize and reorganize as long as all the material has been organized into these objectives. But I think Alison has a great point too. Who is our audience? And I think, one of our key audiences is really other scientists and program managers, funding agencies, the broader scientific community. I think it'd be nice to have the executive summary reach the public and lay people, but I'm not sure that this document necessarily needs to we want to have some granularity so that we can express the ideas.
So in that sense, are we talking about writing these objectives under the context of this structure as we have it now, and then just throwing then just rearranging this structure afterwards? Is that this is the message I'm getting from people? Yes. Okay, because we obviously have to split into groups and do this. Why do we have to split into groups? Yeah.
It's actually last year worked well. We would bring it up in a small group and say, oh, how about this point, and we could talk about it a little bit.
We sat down and we had we were all on our laptops and.
Kristen, if we have like these ideas of things that are, sounds like there's kind of, is it worth having like a little like wild card space on there? So if people are seeing like an emerging theme across that they want to like elevate out, just to add that at the end or something, or space at the end. And that way if folks are starting to get a sense of where the organization might go, they could throw those ideas in there. And then when you guys are trying to deal with this later, that might be a way to kind of get some of those ideas out now.
Yeah, the what are we missing things. And I think, this is comments to be added. I mean, this is it's an initial document. It's not going to be the final, like it says, it's a straw man. So, anybody else? Christoph? Oh, no.
I had 2 comments. One, so I was on the theme group synthesis, and so if that's in good shape, it was actually helpful to have a group together to push forward. So in that sense, it might be helpful to do breakouts. The other thing, though, I do wonder that seems to be missing here, it goes a bit to the structure, but you had that really nice summary from Scott of the implementation, which is like kind of what I was saying, seems like an overarching thing.
Yeah,
right? I mean,
exactly, yeah.
So do we need a 4th group for that to kind of summarize?
I think every group should, my personal opinion on this is that every group should think about the implementation if they're focusing on a topic or a theme.